Stefan, thank you very much for taking the time to speak about these exciting developments that you are heading in Agilent's liquid phase separations division. You just announced the launch of 3 bio-LC systems that were developed to meet the needs for the biopharma industry. Please, can you tell us more about the strategic importance of these new products for Agilent?
Yeah, sure, I can give it a try. So, according to McKinsey, pharmaceutical and medical products sides, pharmaceutical and medical product side, biopharmaceuticals generate global revenues of 163 billion, making up about 20% of the pharma market today. It's by far the fastest growing part of the industry. Biopharma current annual growth rate is more than 8% and is double of the conventional small molecule pharma. And growth is expected to continue at that rate for the foreseeable future. Because of this progressive growth rate, most major pharma companies are really focusing their R&D towards large molecule biopharmaceutical drugs. And the number of biomolecules undergoing clinical trials and the success rate for biologicals has been over twice that of classical small molecule products. As the number of these products rises and new process technologies such as continuous manufacturing are introduced, the complexity of biopharma operations and the biopharma supply chain will increase. Also, current production programs are already stretching the industry, with several players, as we all have seen, failing to deliver to the market. This challenge will only increase as sites are now moving from current one line, one product setups towards more nimble, flexible, multiple product operations. And are required to manage both current and future technologies under one roof. So what sets the Agilent BioSC portfolio apart in this whole game is the depth and width of the complete portfolio. There's also several unique performance characteristics and capabilities such as ISAT, highly differentiated special workflow solution, and maybe we have a chance to talk a little bit more about that in this interview, built on the new core systems, specifically designed columns, supplies, and services, and very importantly, it's really easy to integrate these systems into the existing software environments of the customers, which is absolutely a key to their operations and efficiency and effectiveness. And if I may add just one word very concretely to the new 3 new bio-LC systems. So these 3 systems really will help our customers to find the optimal configuration for their application and environment. So we are not forcing them, so that's the one and take it or leave it. But if I, for instance, need the absolute highest possible separation performance for very highly demanding or complex sample, I would choose the 1290 bio-LC with a high-speed pump, which comes with 1300 bar 2-channel high-pressure mixing. And if I need, for instance, highest possible method flexibility, for instance, for method development or multi-method setups, but also a very high separation power, I would choose the 1290 bio-LC with a flexible pump, which comes with 1300 bar, but 4-channel low pressure mixing capability. And if I work in QC with many different routine applications, one of the 2 1260 bio-LC systems will probably be the optimal choice for me. So yeah, that's initial thoughts on this one.
Yeah, thanks, Stefan. Thank you so much. And as you mentioned, the biopharma market is really one of the strongest growing markets in the field of life sciences. What would you say from your perspective, as you have so many years of experience in the field, what are the biggest challenges in the biopharmaceutical research and development? And also how does Agilent support R&D in this field?
Yeah, thanks for that one. I mean, the growth and investment in biopharma, that we are seeing is also really driving the demand for more innovative analytical instrumentation. And through this launch, Agilent is responding to the challenging analytical requirements that the industry is facing. So when you think of it, developing and reproducing large molecules reliably at an industry scale requires both R&D and manufacturing capabilities of a previously really unknown sophistication. Small molecule drugs typically consist of like 25 atoms. Biomolecules are much more complex and can contain anything from 2,000 to 25,000 atoms. That's so like comparing Empire State Building with a two-story house, right? And along with the larger size of the molecule comes the possibility of many more post-translational modifications and variability of the molecule. The characterization methods must be capable of identifying any potential change that could impact the safety and efficacy of the drug. And the laboratory equipment used to analyze biomolecules must really offer to be secure, must really offer unsurpassed resolution, accuracy, sensitivity, but also speed. We all know time to market is super critical for this industry. So what we would hold central to any decision made about new product development is really listening to our customers. We have heard from customers for many years that they want us to look at a more integrative approach of developing workflows. In the past, we had focused typically on individual products. Now, when products are brought to market, the focus is also on the entire integrated workflow, end to end, meaning how individual products work together and the elements needed for a successful integration and automation. Agilent has put together recommended configurations that are most suitable for a given critical quality attribute characterization and tested methods from A to Z. So for instance, we start with sample prep using Agilent reagents and automation with an SMAP Bravo system, then moving on to separation using the HPLC column designed for CQAs. And then, of course, we also offer the appropriate set of optical and mass spec detectors. And all of that needs to work together, needs to be managed together, and then also has to be finished off with a sophisticated data analysis. So, and has to be supported by, excuse me, a sophisticated services team. So this is what we mean with really A to Z, end to end. And this is really what our customers are looking for, that we help them in the complete endeavor and not just put individual pieces on the table.
Okay, great. Yeah, thank you. So evidently, bio-LC, as you mentioned, is one of the most important trends in LC, and it's definitely an excited market to be in. But in your position, you are also looking at other application areas of LC. Can you tell us more what other developments you have been observing?
Happy to do so. And if you allow, I would like to mention and elaborate on two areas. First, going a little further on this topic of workflow automation in general, and secondly, online process analytics. So for the workflow automation, I refer here to 2D LC, or more generally speaking, multidimensional LC. As a matter of fact, there's customers, not only, but especially in biopharma. But again, it's also in other industries. Who work already today with 3, 4, or even 5-dimensional setups, meaning these really sophisticated systems do not only comprise 2 columns and 2 pumps, but up to 5 columns and 5 pumps. So you may ask, why are customers doing this, dealing with such complex and demanding system configurations? And the answer is relatively simple, because it's worth it. What customers aim to achieve, and I mentioned it briefly before, is really to integrate and automate as many of their manual steps. They have this multi-step assays, and these assays sometimes take longer than 24 hours to prepare and analyze. Integration and automation results in lower variability, meaning higher data quality, higher accuracy and precision, and also significantly faster turnaround times. There is a reduction of total sample preparation and analysis time can easily exceed 80%. So that is really a drastic productivity gain that you can achieve by this kind of integrated and automated workflows. A rather simple example for this, for instance, is the integration automation of two CQA methods for titer and aggregation analysis, which can be achieved via protein A and size exclusion chromatography in one single 2DLC run, which takes less than 10 minutes. Agilent has just released a dedicated protein A SEC kit for such a 2DLC MUM solution, which help our customers to get up and running in this one really fast and conveniently. And we have specifically designed services also that help customers on that one and really make sure that they get what they really need from the systems in terms of performance and data quality. More so, the possibilities for automated sample preparation, which within a sophisticated state-of-the-art UHPLC system can include many more steps, such as denaturation, reduction, alkylation, online digestion, desalting, all of that done prior to, for instance, a peptide separation and detection. And these kind of highly integrated and automated workflow configuration can also include up to 4 or 5 different columns. So that were comments on the multidimensional and automated sample preparation part. And by the way, from our from customer service, we know that 2/3 of our customers consider to go in that direction in the future. And if you allow, I would also like to add comments on the second point, the online analytics. Yeah, okay. Good. So for this part, the online process analytics, Agilent is already working with customers from many major biopharmaceutical, but also here chemical companies since a while. And these customers approached us with a need for better tools, allowing them to do real-time monitoring of critical process parameters, CPPs, and critical quality attributes, CQAs, for real-time release of their biopharmaceutical or also chemical products. Among these critical pain points of our customers in this field are, for instance, the lack of process information, the optimization of the process with regards to yields and turnaround times, and high variability due to manual interaction. There's also an increasing demand in the industry to move towards continuous manufacturing and PAT, process analytical technologies based on process control. So this is certainly an exciting direction. And actually, I'm very happy to also mention that here. We just launched for the first time a dedicated system for automated online process analysis via HPLC applications. We call it the 1260 Infinity 2 Prime online LC system, and this is orderable since June 1st this year. And the heart of this new solution is the new 1260 Infinity 2 online sample manager. That is a complete new device. And together with an also new online LC monitoring software, which coordinates the process sampling and sample analysis, they feature new and proprietary technologies offering customers new process monitoring capabilities. Especially worth mentioning is the fact that the Infinity Lab online LC solutions can be configured with more or less any analytical system configuration and detector, including mass spec. Even an 2DLC online sampling setup is possible. Efforts are also underway to make the bio online sampling system compatible with current bioreactor sampling devices for sterile sampling and filtration. So during the BioLC introduction, we are now already also announcing the biocompatible version of the online LC, which is based on the 1290 Infinity 2 BioLC, and we expect that to be released by end of the year. It is very important for our biopharma customer to analyze multiple attributes as biomolecules are very complex. And the Infinity Lab online LC solution supports exactly this request via a feature called Retain Sample Collection. Yeah, I think that's it. So just giving some highlights on these two areas, multidimensional LC, online sample preparation, and process monitoring, two very important fields where we see a lot of demand and future growth.
Yeah, thank you so much, Stefan, for this insight also and the customer demands that you were talking about. There is one other area I would like to touch, starting with a quote that you might be familiar with. Selecting a column is part science and part art. This is the first sentence of the chapter of your comprehensive ebook on LC that you published, or the chapter on HPLC column selection. And I imagine everyone who has been working in the field would agree with that. On the other hand, and you touched on this already, one of the emerging trends in LC, but also in many other disciplines, is the demand for easy-to-use instruments. And yeah, you already mentioned the integration and automation developments that are going on. Especially important for complex systems to save time. But how can also inexperienced users get a quick start into these very, very complex methods?
Yeah, very good question. And you are, of course, right at the center of it. Ease of use is really critical. You know, ease of use translates into less errors, less mistakes, less downtime, and overall higher data security and reliability of the whole setup. And that is, of course, for the overall productivity of our customers' lab really critical. And I would say ever since the introduction of the, or latest, with the introduction of the Agilent 1100 series in the late 90s or mid 90s, ease of use always has been a central development philosophy of all Agilent LC systems, both for hardware and software. And the good news is that if a user is already familiar with an Agilent LC, they will be able to use the Agilent's new bio LCs without any special training, because these bio LCs are really fully based on our proven award-winning 1260 and 1290 Infinity II systems. And they have the very same set of functionalities and performance characteristics. The only difference is that in addition, the bio-LCs can handle extreme salt and pH conditions to ensure the integrity of the biomolecules during, for instance, as said before, during the automated sample prep, separation, and detection. All of this makes it really very easy for our customers to implement and integrate their new bio-LCs into their existing lab environments. Another important thing is really that you can handle a very broad, basically the complete range of different biomethods on one and the same system. During validation of the bio-LC, we developed several application notes focusing on the CQAs of monoclonal antibodies, for instance, aggregates with size exclusion, charge variance with ion exchange, peptide mapping on reverse phase, and many more. All of those applications are proving the excellent performance and repeatability, but also other biomolecules like oligonucleotides as relatively new biopharmaceuticals in gene therapy, phosphorylated compounds like nucleotides or phosphorpeptides, or in general, iron-sensitive compounds greatly benefit from iron-free flow paths of the bio-LCs. So we have developed application notes for all these workflows to help customers getting started easily and with confidence. And these app notes can be downloaded from the a.com website, where we always offer these kinds of application notes. And if I can add one more thing, we learned during the development of the systems and applications, it is that the universal character of the bio-LC really helps to facilitate daily life in the laboratory. For example, you absolutely have the choice to run your peptide map, then switch to a high salt application like hydrophobic interaction chromatography, thanks to the corrosion-free resistance flow path. And after flushing, maybe you go back to a small molecule analysis, all on one and the same system. With a bio-LC, you really have like a workhorse with the same complete range of configuration choices you also know from our standard Infinity 2 system. So overall, we feel it is really an easy and straightforward switch from our standard systems towards bio. And there's no reason to be afraid of it.
Yeah, that sounds great. Very promising also for people who are new in this area, who are coming from other disciplines. That sounds very helpful. Stefan, again, thank you so much for all these insights, and I can see how exciting you are about these developments. Maybe one additional question. Can you tell us what will come next?
What will come next? I mean, we did pre-announce already some of the products that we will introduce until the end of the year. So what we started with are really what we also talked about today, these 3 new core systems, right? The 2 versions of the 1290 bio-LC, a new 1260 bio-LC, and also the existing. So basically we have 4 configurations for the different needs and environments. And then based on top of that, 2 I mentioned already, we are introducing a bio 2D LC solution, right? So taking our proven 2D LC technology that we started with in 2012 to the bio world, if you will. Then, as I also briefly talked about, the online LC system to support process analytics. Then we have, for instance, this new kit that supports the protein A size exclusion MEM method. What else? We come for when it goes to bio 2D LC-MS, we also have a higher level of integration there, and we will offer compliance services for that one as well. We are going with the online processing into the bio world also. So I think that gives you an idea of the directions we are heading. What we always try to make sure is we want to get our core right, the fundamental systems, and then once we have that in our hands, then we quickly start to put really the more sophisticated workflow solutions on top of it. And that is a general philosophy, and that was very successful in the past, and we think it will also be in the future.
Great. Yeah, thank you so much. I think we are already very impressed about the new developments that you just released, and we'll look forward to all the other things that are coming in the near future. Again, thank you so much, Stefan, and yeah, good luck with everything. It sounds exciting.
Thank you, Christina. It was a great pleasure. Bye-bye.
Bye.